Everyone, welcome to Cell Talk presentation. For today's presentation session, Anshman is a presenter and he will be presenting on the topic OmiEmbed, a unified multitask deep learning framework for multi-omics data. Before the presentation, I would like to introduce Anshman. Anshman Roy is working at Renix Life Sciences as a research intern, focusing on developing machine learning-based solutions to analyze the physiological data and monitor patient vitals. His primary responsibilities include conducting perimetric profiling of health data, utilizing vision language models for data extraction, integrating optical character recognition to digitize and process textual medical records, and analyzing medicine images data to enhance diagnostic accuracy. Anshman is a pre-final year undergraduate student pursuing a Bachelor of Technology in Computer Science and Technology at the Indian Institute of Engineering Science and Technology, Shivpur, maintaining a CGPA of 9. His academic experience includes research internship at Internal Institute of Information Technology, NAYA Raipur, and IIEST Shivpur, where he worked on machine learning models for health data analysis and algorithm optimization. Throughout his studies, he has developed a strong interest in artificial intelligence, machine learning, and full stack development. Anshman has acquired hands-on experience in developing AI-driven solutions and performing data analysis. He aspires to advance his career in the domain of artificial intelligence and machine learning. Now I welcome Anshman to start his presentation.
So good evening, everyone. So today my topic is OmiEmbed, a unified multitask deep learning framework for multi-omics data. This paper was initially published in the journal Cancers, which has an impact factor of 6.575 in the year of 2021. Now, let me start by introducing this topic. Now, high-dimensional omics data, such as gene expression, DNA methylation, and microRNA expression, holds critical biomedical information for personalized medicine. However, these datasets come with an overwhelming number of molecular features. For example, over 60,000 plus identifiers when it comes to RNA sequences, nearly 485,000 probes for DNA methylation, paired with relatively few samples. This imbalance creates what is known as the curse of dimensionality. Traditionally, researchers have relied on manual feature selection or standard dimensional reduction techniques, but these methods can miss complex relationships hidden in these data. Now, before we discuss the framework, let us review the omics data types that are involved. Now, here we have gene expressions, which are RNA sequences, which have around 60,000 identifiers. We have DNA methylation, which are approximately 485,000 probes. And we have microRNA expression, which are around 1880 identifiers. Traditional methods have depended on manual selection or statistical approaches, such as principal component analysis. In contrast, earlier deep learning approaches typically focused on single omics types or single tasks. They lacked the ability to integrate different data types and perform the multiple analysis simultaneously. Now, we have many present challenges. One present challenge that I just discussed was the curse of dimensionality, which leads to models being overfitted. We also face complex and non-linear relationships among molecular features, which are difficult to model. Also, multiple analytics tasks such as classification, regression, and survival prediction often require separate methodologies. Integrating diverse omics data types into one coherent analysis framework is inherently complex. There was clearly a need for a unified solution that could address these challenges into a single model. So the main objective of this research was to develop that unified model. A unified multitask deep learning framework, OmiEmbed, was developed for that. It reduces the dimensionality of high-dimensional omics data by embedding it into low-dimensional latent spaces. It integrates multiple omics data types, including RNA sequences, DNA methylation, and microRNA, simultaneously performs several downstream tasks, such as dimensional reduction for visualization, tumor type classification, demographic and clinical feature reconstruction, and survival prediction. It leverages multitask learning to achieve improved performance over training each task separately. Now, coming to the methodology, OmiEmbed is built around two core components. One is the deep embedding module, and another is the downstream task module. The deep embedding module, this module is based on variational autoencoders, VAEs. It compresses the high-dimensional omics data into low-dimensional latent vectors. This framework support multi-omics types with specialized encoders. For example, for gene expression, DNA methylation, and microRNA expressions. They also provide the option to use either CNNs or fully connected neural network structures after we have gotten the latent space representations. And then comes the downstream task modules. The downstream task modules are designed to do each specific tasks, like for classification tasks, for regression tasks, or for survival prediction tasks. Now, let me explain this architecture of OmiEmbed in a little bit more detail. Now, here, initially we have the encoders. Now, as you can see, the encoders take in this information, then they encode it into smaller latent spaces. And then they decode with that particular information, that original information. And then using reconstruction laws, this cycle goes on and they learn that latent space representation properly. It is, like giving an example, it is kind of like summarizing a book effectively. Like suppose I have like a 100-page book and I just want to summarize it in 2 pages. This encoder-decoder structure does the same. It finds the exact summary that is needed. The summary that can effectively have all the information represented in much lower space. Then after that latent space representation has been learned, we have each individual heads for each individual tasks, like a diagnostic task head, for a prognostic task head, or a demographic task head. Now, coming to the training methodology. Now, training here happens in 3 phases. The phase 1 is an unsupervised pre-training of the embedding modules. Here, the VAs, the variational autoencoders, learns to compress high-dimensional omics data into lower-dimensional latent space. It does this by minimizing the reconstruction loss plus a KL divergence regularization term to ensure that latent space captures the key features of the input data. Then comes the phase 2, that is the downstream task training. Once the latent space representation is established, the model fixes the embedding and trains the downstream modules. Example of like a fully connected neural network for classification, regression, or survival prediction, using latent space vectors as its inputs. This step optimizes each task individually based on its own specific laws. Then we have the last and the most important step, which is the end-to-end fine-tuning. Now, finally, the entire network, both the deep embedding module and the downstream task module, is fine-tuned together with a joint loss function. This step refines the latent representation to better serve all downstream tasks simultaneously. Now, let me actually explain this with a particular example. Now, let us imagine a gene expression profile from a patient where each sample has like 60,000 gene expressions values. Like we can think of it as like a long list of numbers. Now, what the latent space representation is that the VAE encodes this like 60,000 dimensional vector and compresses into a much smaller vector, say just 128 dimensions. Here, the dimensions is that like there were 60,000 values and I'm compressing that 60,000 values into like 128 values. Now here, each of these 128 numbers captures the blend of information from these thousands of genes. At the same time, what the decoder tries is that it tries to reconstruct that original 60,000 list from that 128 dimensional vision representation. So here 2 things are involved. One is reconstruction loss, the difference between the original profile and the reconstructed one. It tells the networks how well it can learn its data effectively. And there is also something called a scale divergence, which tries to smooth this graph into something which is closer to a Gaussian curve. Now, after we have learned this representation, in the phase 2, what we do is that we just freeze this encoder part and we give that data and it gives us the latent space representation. From that latent space representation, we train a normal fully connected neural network in the normal way that we do. And that helps us to train these individuals task-specific heads properly. And at the third and the last stage, what we do is that we combine this entire process and unfreeze all the what can I say, everything, and all the weights and biases, both of the encoder and of the particular head, gets trained simultaneously to fit that particular task properly. Let me go to the next. Here, like the paper has dealt with 2 different data sets. One is GDC pan-cancer multi-omics dataset, and another one is a GAC 109381 brain tumor methylation dataset, also known as the BTM dataset. In the first dataset, this is a comprehensive dataset formed by what can I say? Formed by multiple expressions. Here there are RNA sequence gene expressions, there are DNA methylation expressions, and also there are microRNA expressions. In the second data sets, comprises of like 3905 samples covering the WHO-defined central nervous system tumor entities using a 450K DNA methylation arrays. Now, coming to the results, let us first go over the dimensional reduction thing that is done by the first training step. The OmiEmbed effectively compresses the higher dimensional omics data into 128 dimensional latent vectors. Using something called as TSNE visualization of the BTM dataset, the unsupervised phase reveals clear clustering of different brain tumor types. Like, let me show this by this image. Like, this image shows clear clustering. This clustering indicates that the latent space representation that the model has learned is proper. Now, coming to tumor classification. When it comes to tumor classification, OmiEmbed outperforms 8 other models in the BTM dataset. It achieves 83.2% macro F1 score and a macro TPR and a macro PVR around 84%. The overall accuracy is around 87.5%, and the macro ROCUC reaches around 99.4%. On the GDC dataset, the best performance was obtained when integrating all 3 omics types, reaching 97.7 accuracy. This highlights the power of multi-omics integration for precise tumor classification. As it is visible from this particular bar chart, the particular approach outperforms all other approaches. Also, let us come to next for the age prediction. Now, the age prediction in this particular thing is tackled as a regressive task. OmiEmbed demonstrates superior performance with a mean absolute error of 6.93. Sorry, with a median absolute error of 6.93, a mean absolute error of 8.37, and an RSME of 10.66, and an R-square score of 0.479. It shows, these results shows that were significantly better compared to the traditional dimensional reduction methods combined with regression or a deep neural network regressor. This shows that the model can extract age-related molecular patterns effectively. Now, coming to the most important and the difficult part was the survival prediction. In survival prediction, OmiEmbed achieved state-of-the-art performance. A coordinates index, C index of 0.772 and an IBS of 0.166. It outperformed 9 other methods, including traditional and deep learning approaches. The 3-phase training strategy is clearly reflected in the learning curve with distinct transition between phases. The survival module outputs personalized survival cards, providing not only risk scores, but a complete risk profiling over time, which is crucial for personalized treatment planning. Now, here, it is showing the learning curve of 3 different stages. As it can be seen, the last step contributes heavily to reducing the survival loss and training the model very effectively. Now, coming to personalized survival curves, one of the standout features of this particular model, OmiEmbed, is its ability to generate individual survival probability curves. For each patient, the model predicts the probability of survival over time, effectively capturing patient-specific trajectories when compared to actual outcomes. The prediction shows a strong correlation, offering clinically actionable insights that can perform personalized treatment decisions. Now, here, this particular curve is the profiling that they have done, and these particular lines show the death time of those particular individuals. As you can see, that curve effectively represents when it goes down and the death time being close to it. This shows that it is able to model these personalized curves properly. Now, the most and the best part of this model was the multitask learning. When, after the third step in the multitask learning, the C index improved from 0.772 to 0.782. Tumor classification F1 score increased from 0.952 to 0.965. And the age prediction, the median error decreased from 6.93 to 6.98 years. Sorry, 6.93 to 6.68 years. This shows that the model, when trained on multiple tasks simultaneously, learns from each individual task properly, which helps the other tasks too. Now, coming to the latent space analysis, the TSNE visualization of the latent space, colored by the tumor types, age, and risk score, demonstrate that a single embedding captures clinically relevant factors, further supporting the biological interpretability of learned representation. Here, this thing shows properly. This is something in machine learning we call as like almost perfect clustering. Like after the third step, they are able to cluster everything in a very perfect manner. This shows that the latent space representation after the final phase of tuning is almost perfect. Now, Coming to the summary, OmiEmbed outperforms state-of-the-art methods across all evolution tasks, including unsupervised clustering, tumor classification, feature reconstruction, and survival prediction. The unified multitask framework not only integrates multiple omics types, but also improves performance over individual task training. It provides comprehensive molecular to phenotype mapping that is essential for precision medicine. Now, coming to the advantages, the first and the most important advantage, it is the first unified framework for multi-omics tasks. Being the first unified framework for being able to capable high dimensional data and multi-omics integration, classification, regression, and survival prediction into all in a single model. It also shows superior performance due to multi-task learning strategy, which enables better overall accuracy and efficiency. It is practical clinical implications such as more precise tumor classification and personalized risk stratification, which can direct impact treatment decisions. Now, coming to the limitation, the most important limitation is that the computation complexity for large datasets is quite high, which requires a high GPU power. And it is also limited to specific omics types currently. The future works can be adding more omics types, integrating to clinical imaging data, like transfer learning between different cancer types, application to single-cell multi-omics data, and also explainable AI components for clinical interpretations. So the final conclusion is that OmiEmbed is a powerful unified framework for multi-omics analysis. Successfully tackles the curse of dimensionality challenge. It performs multiple downstream tasks simultaneously with better performance, demonstrates that multitask learning for omics data is, and also the entire thing is open source, so that anyone can take their code and use it for any downstream tasks and can improve on it. It is also highly adaptive to various omic types and downstream tasks, and has potential to facilitate more accurate and personalized clinical decision making. Now, the impact of this research is very wide. It has significant impact when it comes to cancer precision medicine. It has a significant impact when it comes to computational biology and also to various clinical applications. Thank you. That is it for my presentation.
Thank you, Anshuman, for the nice presentation. Audience, now the floor is open for Q&A session. If you have any queries, please type them in the chat box. So, Anshuman, we have a question from Vijay sir. Is there any biologically significant discovery made using this framework?
The paper doesn't actually talk about it in that particular manner. This is more like on the lines of like an advancement towards the machine learning in this particular domain. There isn't any biologically significant discovery, but what I have seen is that the ability for this model to actually train and completely separate kinds of data and still able to learn about the latent space representations better using different kinds of data and improving each other's accuracy is a significant thing that has been showed here. That can be applied to many other fields, and that can really improve many, many detection and many prediction accuracies.
Yeah, thank you, Anshuman, for answering it. And that's it. I think we don't have any more questions. So this will be the end of the session. I thank Anshuman. It is very informative. And I thank everyone for attending the session. And I hope to see you all again in the next presentation. Thank you.
Thank you.